Hey guys, welcome back to my channel. So today's video is going to be on protein synthesis again. So this is for EDEXCEL GCSE this time, and this is for all specifications, so for biology foundation and higher, and also combined sciences. So let's get into the video. So the first two specification points are included on all of the EDEXCEL GCSE specifications. So these are to explain the importance of enzymes as biological catalysts in the synthesis of proteins and their breakdown into amino acids, and to describe a gene as a section of a DNA molecule that codes for a specific protein. For our first specification point, we're going to be discussing enzymes, and so here we have our enzyme, and enzymes are known as biological catalysts, and these enzymes are really important in catalyzing protein synthesis and also protein breakdown reactions. So here we have our amino acids and our enzyme, and the enzyme is going to help us in protein synthesis. So it's going to help these amino acids bond together to form new proteins, and then when these new proteins need to be broken down back into amino acids, this is where the enzyme helps too. Moving on to our second specification point. Here we have our cell and our nucleus, and within the nucleus is our genetic material, our DNA, and a section of DNA is known as a gene, and this gene has a specific code of bases, and this code determines the sequence of amino acids, which then influences the specific protein. So if we were to put that into a definition, it's just as the specification point says, it's that a gene is a section of DNA that codes for a specific protein. So now we're going to move on to some questions based on that information. So the first question is, what are proteins made of? And then we have some answers. So we are obviously aware that proteins are in fact made up of a sequence of amino acids. So our answer is therefore A, amino acids. Question 2, then we need to explain what a gene is. And we know that a gene is in fact a section of DNA which has a specific code that codes for a specific protein. So here we have our answer. So gene, a section of DNA that codes for a specific protein. Question 3 is from OCR GCSE Combined Sciences B Foundation Tier sample paper. And the question is, scientists use key terms to explain genetics. Use words from the list to complete the sentences about genetics. And the sentence is, the molecule which carries the genetic information is DNA. DNA is a polymer made up of blank. Sections of DNA which code for a particular protein are called blank. So we know that in fact DNA is a polymer made up of nucleotides, and don't worry if you don't know this, I will be doing a video on DNA and RNA as well, so we don't need to worry about that part for the specific information we've just learned. But for the second part, sections of DNA which code for a particular protein are called, we know that that is in fact genes. As a gene is a section of DNA, it has a specific code, and this codes for a particular protein. So there we have our answer. The next set of specification points are what you need to know if you're on the EDEXCEL GCSE Biology higher tier. And these points are, number 1, to explain how the order of bases in a section of DNA decides the order of amino acids in the protein. Number 2, describe the stages of protein synthesis, including transcription and translation. So for our first specification point, here we have our DNA and our gene selected. And our gene is of course a section of DNA as shown there. So a gene is made up of bases. So the bases within DNA are A, T, G and C. So a gene has a sequence of bases shown here, and it is in fact a triplet code which determines the order of amino acids. So a 3-base sequence is the code for a particular amino acid. So as you can see here, so 3 bases here, TAC code for this particular amino acid, then CGA coding for this particular amino acid, and so on and so forth. So now for our second point, which is in fact describing protein synthesis itself. So it's the stages of transcription and translation. So protein synthesis actually begins with the process of transcription, and this occurs in the nucleus. So here we have our DNA again and our gene, and the DNA does in fact unzip or unwinds around the gene. And this causes the DNA strands to separate. So there's 2 DNA strands. One is known as the coding strand, and the other one is known as the non-coding or the template DNA strand. So after the DNA strands have separated, RNA polymerase, which is an enzyme, binds to the non-coding DNA. So it binds to the template DNA strand, which is located in front of the gene. So after the RNA binds to the template strand, an mRNA strand, which is a copy of the gene, is actually produced. So this is produced by the RNA polymerase, and it is a complementary mRNA strand, which is produced from the coding DNA of the gene. So just a reminder that for the bases, DNA is made up of A, T, C and G, and RNA is made up of U, A, G and C. So once the mRNA strand is finished being formed, it actually moves out of the nucleus through a nuclear pore, and then we move on to translation. So translation occurs in the cytoplasm, and once the mRNA comes out of the nucleus, it actually attaches to a ribosome in the cytoplasm. So one of the key structures of mRNA is that it's made up of codons, and these codons are triplets of bases. And it is in fact these triplets of bases that code for specific amino acids. So for example, AUG codes for methionine. You don't need to remember this, but you do need to remember that the triplet code is a key factor of mRNA. So then we come to the molecule tRNA, which is also known as transfer RNA. So this molecule has an anticodon at the bottom, and it also carries a specific amino acid at the top. So during translation, tRNA actually transfers amino acids to the ribosome. So as tRNA continues to bring amino acids to the mRNA at the ribosome, amino acids are linked by peptide bonds, and this is what eventually forms the polypeptide chain, which is the first structure of a protein. Then I'm going to move on to some questions based on that information. So our first question, which comes from OCR GCSE Biology A Higher Tier Paper 1, also referred to as Paper 3, and this is from 2018. So our question reads, during protein synthesis, the DNA that codes for a particular protein is copied. This copy is called mRNA. The diagram below shows the base sequence for a section of DNA. And our question is, what would be the complementary sequence of mRNA for this section of DNA? So we know that our DNA bases, as shown in the base sequence, are G, C, T, and A. But for RNA, we have G, C, U, and A. So our RNA includes U base, not T. So if we have a look at our answers, we can see that A and C include T bases. So we know that these definitely can't be the right answers because mRNA and RNA itself do not contain T bases. So we can discount A and C. And then if we have a look at our actual sequence, so we're starting with G and then another G. So if we have a look at B, so we've got C, C. So C does complementary base pair with G, so we know that is correct. And then if we look at D, we've got G. So we know that G does not complementary base pair with G, it's in fact C. So we can there discount D. So without even going through the whole sequence, we know that our answer is B. And if we want to go through that to check it, we can have a little look. So we've got G, which goes with C, G, which complementary base pairs to C, and then T goes to A, and you get the picture. So we know that our answer is in fact B. Question 2. So this is from OCR GCSE Biology A Higher Tier Paper 1, also Paper 3, which is a sample paper. I just want to say as well that I know that these questions are from the OCR specifications. However, EDEXCEL didn't have any protein synthesis questions in their papers, so I've taken these from the OCR papers because the specifications are very similar, so just for some practice. If you head over to my website as well, I have put up loads of questions for all the exam boards for GCSE and A Level Biology for protein synthesis. But anyway, let's get into the question. Insulin is a protein made of 51 amino acids. How many bases are in the length of DNA coding for insulin? So we know that DNA coding does in fact have a triplet code. So 3 bases code for 1 amino acid. So basically all you need to do is times 51 by 3. And so our answer is going to be 153. Question 3, which is from OCR GCSE Biology A Higher Tier Paper 2, also referred to as Paper 4, from 2018. So our question is, switching off the protection mechanism involves the plant making a protein. So it does say described in Part B. If you do want to have a read of Part B, that is over on my website in the question pack section for EDEXCEL GCSE. However, I know that it says it in this question, but you don't really need to know about what it says in Part B to actually answer the question, basically. So scientists have put extra copies of the gene for this protein into the plant. So this makes the plant make more mRNA molecules. And the question is, explain why making more mRNA will switch off the mechanism faster. So this is a question which is more about applying your knowledge of protein synthesis. So it may seem a little bit tricky at first. So let's just highlight some of the key parts of the question. So we've got a mechanism here. When it is switched off, so switch off a mechanism, and this involves the plant making a protein, so they've put that in bold for us as well, so we obviously need to highlight that one. And so scientists are putting extra copies of the gene for this protein into the plant, so this is causing the plant to make more mRNA molecules. So we've got more mRNA because of more genes. So explain why making more mRNA will switch off the mechanism faster. So we know that mRNA obviously carries the code for a protein. So if there's more mRNA, then more mRNA is going to be translated, and so more proteins are going to be produced. So based off what it's saying, so switching off the protection mechanism involves the plant making a protein. So if more proteins are being produced, then that means that the mechanism is going to be switched off faster. So yeah. Question 4, which is from OCR GCSE Biology B Higher Tier Paper 1, also referred to as Paper 3, from 2018. So the question says, a mayor reads an article in a magazine which explains that genes code for the production of a taste receptor on the tongue. Taste receptors are proteins. Complete the sentence to describe how a protein is made. Use words from the list. Each word can be used once, more than once, or not at all. So then we've got a selection of keywords there. So the key part in this question is obviously how a protein is made. So we know that this is targeting protein synthesis itself. So we've got transcription and translation. So it's likely that this passage is going to explain both of those stages. So if we look at the first sentence, so a copy of the blank is made from blank. So we know that initially at the beginning of transcription, we have a section of DNA which is known as a gene, and a copy of this gene is made RNA nucleotides form mRNA. A copy of the gene is made from mRNA. We then have our second sentence. So this molecule travels to a something in the cytoplasm. So here we've got the mention of cytoplasm, so we know that we've moved on to translation. So this molecule being mRNA travels to a ribosome in the cytoplasm, because this is where protein synthesis takes place. It is on the ribosome. So then our next sentence, so here blank are joined together to form a protein. So we know that once the mRNA reaches the ribosome, tRNA molecules bring amino acids to the ribosome. Amino acids that bond together in order to form our polypeptide, our initial structure of our protein. And then finally, we've got a sentence about mutations, and I haven't covered it in this specific protein synthesis content. But just for the sake of completing the question, we've got a mutation will create a something and therefore a different receptor. So a mutation is obviously going to change the amino acid sequence and therefore change the protein. So a mutation would create a genetic variant. So here we have our answers. Question 5, which is from OCR GCSE Biology B higher tier sample paper. So RNA is another type of nucleic acid. It is involved in protein synthesis. The table shows the nucleotide sequence in RNA that codes for different amino acids in protein. So we've got our amino acid, then we've got the amino acid abbreviation, and then the nucleotide sequence. So it says, look at the nucleotide sequence in X section of RNA. Write down the sequence of amino acids that this section of RNA codes for. Use the abbreviation for each amino acid. So if we have a little look down the sequence, so we've got GAC, so we know we haven't got GAC here. ACU, no ACU here, CUG, oh okay, we do have a CUG which is here, so that's there, so that must be our first triplet code, our first sequence of 3 bases. Therefore leucine is our first amino acid, so since it says to use the abbreviation, it must be Lu, so LEU. And then we go across again, so then we've got GAG, so GAG is here, which codes for glutamic acid, so GLU is our abbreviation. And then we've got UAC, so UAC is here, so that is tyrosine, so TYR to go in here, and then we've got ACG, which is here, and that codes for threonine, so THR to go in the final box here. So then we've got our answer there. Question 6, which is from OCR GCSE Biology B higher tier sample paper. Antibodies are proteins. The statements below describe protein synthesis. A copy of the gene is made from messenger RNA. B, the gene that codes for the protein is found in the DNA. C, the mRNA travels to a ribosome in the cytoplasm. D, the ribosome joins the amino acids together in the correct order. And then it says put the statements in the correct order. So we know that the first stage of protein synthesis is transcription and the second is translation. So it's evident that we've got 2 statements on transcription, A and B. So B, the gene that codes the protein is found in the DNA. So this is going to be our first statement. This is the first thing that happens in protein synthesis. And then A, a copy of the gene is made from messenger RNA. So then we know that RNA polymerase brings the RNA nucleotides to the template strand, and the base is complementary base pair, and then the messenger RNA strand is formed. And this is a copy of the gene. So we've got B, then A, okay, and then we've got C. So the mRNA travels to a ribosome in the cytoplasm. So this is the beginning of translation, and then the ribosome joins the amino acids together in the correct order. So that's obviously the final stage of translation when the polypeptide is produced. So here we can see our answers. Now I do want to say that when I actually got this question from the sample paper, they've actually made a mistake in the answer and they've put the wrong sequence of letters basically, so they've probably adjusted the question, changed the statements around, and they haven't changed the answer they've put. So if you do have a look at this question, don't get confused. They've basically made a mistake and this is the correct order of statements. So thank you so much for watching, guys. Make sure to head over to my Instagram page and give me a follow. Also, if you've got any video requests, make sure to comment them down below or DM me on Instagram. Please subscribe to my channel and like and share. And also head over to my website to access loads of exam questions and revision resources on protein synthesis for GCSE and A Level, as well as other topics as well. All right, thank you. Bye, guys.